Dr. Sturla, we have one question about antibiotics in chicken farming. Do they have an adverse effect on the human gut microbiota if we consume such chicken in our diet?
Yeah, this would be a. Oh, can you hear me? Okay. Yes.
Yes.
Sorry, there was an echo. This would probably be a good question for the for my biotechnology collaborators on the project. I haven't seen. I don't know the data in particular from that, and I guess the main exposure question that I'm not sure about is what is the. I mean, certainly, if humans take antibiotics, it's going to have a well-known effect on our microbiota. But I don't know about the transfer from the consumption of animals that have been given antibiotics.
Yeah, we seem to have a lot of questions about diet and the microbiome. Another question was about consumption of probiotics. For example, L. shirota strain. Does it affect or alter the acrolein cycle or balance?
Yeah, this is a really good question. I didn't express it specifically, but this is really the implication of these supplementation experiments that we did on the microbial communities, and we're doing this direct supplementation in a fermentation model, and then whether you can take that a step further and think about the supplementation in a human is a step that really requires some engineering and know-how on the microbe side. I think my overall response would be that I think there are a lot of examples of probiotics, say proposed probiotic strategies out there that actually don't work very well. So the complex community of the microbiome has a way of reestablishing itself in a way that certain probiotics actually don't have an effect. There's also the concept, for example, of using prebiotics to engineer the microbial community, and so this approach involves altering basically altering the substance that the microbes favor, but certainly there can be the concept can work, and we haven't gone further in testing that in the system that I presented, but it is true that this is going on for other purposes because of the beneficial effects that have been observed with ruterin producing bacteria. And so there are many studies underway that are looking at the potential supplementation of these GDH proficient strains. And so we should be we should see the results of that, and we should see if this is a viable strategy in this case. And then, of course, this brings up the question we have on whether this can be an overall beneficial or if there are actually safety concerns associated with that. And I think the answer to that is going to be it'll probably really depend. It can depend on individuals. It can depend on delivery strategies and really all of the things that go into engineering the microbiome. And again, the biotechnologists who we collaborate with, this is really a topic that they are very skilled at and are working to advance.
Thank you, Dr. Sturla. We had another question regarding, have you used acrolein scavengers such as polyphenols like doxorubicin, which can trap acrolein chemically?
So basically, that's doing the same. The FIP is kind of doing the same thing. It's trapping it, and so we actually, in a lot of the studies, we're using the FIP. At the beginning, we were using the heterocyclic amines because we were focused on understanding the impact on their toxicity. But then in follow-up studies where we're interested in looking more at this supplementation question and looking into the microbial communities, we're really using it then as a tracer molecule. And so we use a mass spec based analytical method to measure that. We've also scavenged it with glutathione is another way to scavenge it. And so we haven't used the particular compounds that are mentioned by the person answering the question, but we've done it either with the heterocyclic amines or other hydrocyclic amines that I didn't show on the slide, or glutathione does it pretty well.
Thank you, Dr. Sturla. And the question, are these heterocyclic amines only restricted to animal-based meats, or plant-based products can also produce it? And second question is, have you checked for short-chain fatty acids in the fecal samples?
Yeah, this is a short-chain fatty acid production is part of the, there's a lot of kind of basic metabolite profiling that goes on in these studies that looked at the complex microbiomes and looking at the relationship between the GDH activity of the microbiomes, different taxa, and their capacity to catalyze the transformation. So I really focused on this HCA transformation because it was related to toxicity, but we have profiles of things like short-chain fatty acids and other standard metabolic processes in there. So keep an eye out for Alejandro's papers that will be coming from his recently defended PhD.
All right, we look out for your tweet on that, Dr. Sturla.
Yes, absolutely.
We have several more questions, but I'll pick and choose a few in the interest of time. There was one more question on the decreasing levels of glutathione. So what is the reason for the fall in glutathione levels? And have you checked some genetic activity around that? And also, what method did you use in your cell viability tests?
So I'll kind of go backwards in my answer. So for the cell viability, we use a couple of different standard assays. I had a couple of different slides with cytotoxicity data, and so we use assays like cell titer glow and different different assays for measuring cytotoxicity that are based on kind of standard in vitro cell-based cytotoxicity measurements. And we often like to even use a couple of different ones so that we really get a good sense of characterizing the system. And then the question is, what is going on with the glutathione? And this is maybe related to my previous answer about the scavengers. And so the glutathione is reacting directly with the acrolein. So glutathione can do the same sort of conjugate addition chemistry to the glutathione. And so then that's a basis of promoting reactive oxygen species that the glutathione might be normally involved in diminishing. I think I got all of the three parts. Yes. I should open. Yes. I have the Q&A here too.
All right. So, Dr. Sturla, I see a lot of questions related to diet and microbiota. Maybe the audience can reach out to you directly on a few of them. And just one last question from us on terms of the journal, and because we did talk about a lot of chemical transformations, an interesting piece of research that often comes to us is about environmental toxicology, nanotoxicology. So could you share some insights on what are the expectations from the journal in these topics, so our audience will have a better idea of what to submit to your journal?
Yeah, I think one of the important features of papers that are reviewed well in the journal is that whatever the exposure agent is that is being studied is typically very well characterized. So you mentioned nanomaterials in particular, and so there should be strong material characterization data that has to do directly with the materials that were being used in the studies. Where, for example, natural products, often these are present as mixtures, and we often like to see studies that that understand the basis of how mixtures are giving rise to biological effects. So often this can mean, for example, testing individual components of the mixture, or really well characterizing the mixture, and then having effects not only of the mixture, but really looking at individual components and really trying to understand how discrete chemical structures can give rise to the biological responses. I think another, maybe a last key aspect that I'll mention is really this mechanistic, this underlying concept in terms of mechanism and understanding chemical, biochemical, molecular level transformations. And so, so if a study makes an observation of a potentially toxic effect, but is a little bit of a black box, we don't know what is happening or what is the basis for that. These studies often have a difficult time of making it very far in the journal, because we're really interested in promoting this mechanism-based understanding of toxicity, and how is it that how are particular biological reactions giving rise to responses. And maybe as an example, the science that I showed today, there's a lot of questions that have to do with the potential health implications and the probiotic effects and strategies, and we're really trying to break that down to understanding these very specific chemical reactions that are happening. For example, with this ruderin-producing bacteria. It wasn't known before that actually acrolein is the acrolein is generated in this system, and that this is a base. It's absolutely required that the acrolein is generated for some of the effects that have been observed for a long time. And we were able to delineate that that's because of interactions with the acrolein molecule. But sometimes it's more on a biochemical level and a molecular level, but really having this link between biomolecular interactions that are then driving the biological observation.